All right, guys, it's 10 o'clock, so we're going to get started. Thank you guys for being here today. Someone please unmute and let me know that you can hear me and that you could see my screen until I just turned it off.
Yes.
Very good, very good. Well, we're going to get started today. We're going to be talking about hypothesis testing and chi-square analysis. So a lot of stats. So I'm going to reshare my screen. And if you guys will, during the presentation, just mute your mics and turn off your cameras. That'll make the Google Meet run better. And then if you have a question, feel free to unmute and ask. We want you to ask if you have questions. And if at any time my screen happens to go out, make sure you let me know. Last week, the screen share stopped about 10 minutes before it was over. So I was talking about things you guys couldn't see. So make sure you let me know if the screen share goes out. All right, so here we go. We're going to be talking about hypothesis testing today. So this is a procedure used by scientists to either reject or fail to reject a statistical hypothesis. We can't really ever necessarily prove a hypothesis, but we can figure out how reasonable it is. And there's really two ways we're going to do that, two types of statistical hypotheses we're going to use. One of them is called the null hypothesis. And I hope you guys know that the word null means none or zero. We'll talk about how that factors in a second. But the null hypothesis is symbolized with an H and a zero subscript, or sometimes we call that H naught. So this is the hypothesis that there's no difference between the two groups of data that you're comparing. Or oftentimes it's the hypothesis that whatever your independent variable was, that it had no effect on the outcome. Oftentimes what you're trying to do is disprove a null hypothesis and therefore give some proof or some evidence to back up your alternative hypothesis, which we're going to abbreviate with H sub A. So this is essentially another hypothesis about why things happen, why there are differences between your two groups, or what changes your independent variable caused. So one of the ways that we're going to test these hypotheses is with what's called a chi-square test. Sometimes you'll hear this called a chi-square goodness of fit test. So this is a test that's used to compare a set of observed data to a set of expected data, what you think you should have gotten based on your hypothesis. The test can be used to reject a null hypothesis, or some people like to say accept a null hypothesis. I think statisticians prefer us to say fail to reject. And we'll talk more about what that means as we work through some examples. But again, remember the null hypothesis is essentially the idea that your two groups of data, your observed, the data you got from an experiment, and your expected, the data you expected to get, are not statistically different, or that your independent variable had no effect on the outcome. Oftentimes on AP Biology FRQs, free response questions, they're going to ask you to write out a null hypothesis. So essentially you can say there's no difference between group A and group B. Or you can say the independent variable had no impact on the outcome of the experiment. It's really all it takes to write a null hypothesis. If your stats, if your chi-square causes you to reject the null hypothesis, this is going to give strength to your argument for the alternative hypothesis. For example, if you're testing the effect of a fertilizer on plant growth, you might start off with a null hypothesis that this particular fertilizer has no effect on plant growth. You're then going to have a set of plants grown with the fertilizer and a set of plants grown without. And you're going to use a chi-square to compare those. And your chi-square is going to let you know if the fertilizer had an effect or not, essentially. And we'll talk about how that works. One thing to remember about chi-square, it can only be used with what's called categorical data. So this is typically data in the forms of counts, like you're counting a certain number of things, or percentages. It's not used to compare means or averages or anything like that. So chi-square used to compare categorical sets of data, usually in the form of either counts or percentages. So I think the best way for you guys to understand chi-square testing is for us to work through some examples. What you see on the screen here are formulas that you're going to find on your AP Biology formula sheet, which you can use throughout the AP exam. This is the formula to calculate what's called the chi-square statistic. Chi is a Greek letter that looks an awful lot like a curvy X. So this is chi-square, you see right here. This formula I know looks a bit intimidating. It's equal to the summation of O minus E squared over E, where O stands for your observed data and E stands for your expected data. We're going to work this out in a table form, which makes this formula very easy to use. We're also going to need to use this table, which is also on your AP Biology formula sheet. This table is going to allow you to find what's called a critical value. These are critical values, which you can use to compare to your calculated chi-square statistic. And we'll talk about how that works here in just a minute. Let's go ahead and talk about an example. This is a common example that biology teachers like to do with their students. So supposedly the M&M company, when they color M&Ms, they're supposed to come out of the factory in a certain ratio. These numbers that I have here are not the real distributions. I just made these up. But there's a formula that they use when they're coloring the M&Ms. So let's pretend that the M&M company says that 30% should be red, 20% brown, 10% yellow, 20% green, and 20% orange. And I'm going to write these numbers down so that I have them in front of me on the next slide. So 30 red, 20 brown, 10 yellow, 20 green, and 20 orange. So then what your teacher would do in this lab scenario is that they would give you some M&Ms and you would open them up and essentially count them. Remember, chi-squares for comparing count type data. So we open up a bag and we count 50 red, 60 brown, 30 yellow, 40 green, 20 orange. These are our observed data. This is what we got when we actually did the experiment, or in this case, just simply counted the M&Ms. And then we can use these percentages up here to calculate our expected values. We'll look at that in a minute. Before we start, though, notice that we're asked to state a null hypothesis. Remember that a null hypothesis is there's no difference between your observed and expected data. So we might say something like, there's no difference between the distribution of M&Ms in the bag and the expected distributions published by the M&M company. Or we could simply say there's no difference between the observed and expected values. That would work too. Next, we're asked to perform a chi-square analysis on this data. So we've got to do some calculating. All right, so I've already done the work here in a table. So let's talk about what I did here. So this set of numbers here in the O column, those are the observed data. It's what you got when you did the experiment or you counted, in this case, the M&Ms. Simply copy those off this previous slide. 50, 60, 30, 40, 20. All right, then we needed to calculate our expected values. To do that, the first thing we need to know is figure out how many M&Ms we have in our bag. So we're going to add up this column. So 50 plus 60 is 110, plus 30 more is 140, plus 40 is 180, plus 20 is 200. So 200 total M&Ms. And then our problem gave us these expected percentages. So to calculate our expected values, we're going to take the total number of M&Ms, 200, and multiply them by those percentages. So for red, that percentage was supposed to be 30%. So 200 times 0.30 gives us this 60. For brown, that percentage was supposed to be 20%. So 200 times 0.20 to get 40. For yellow, that percentage was 10%. So 200 times 0.10 to get 20. For green, it was 20%. So 200 times 0.20 to get 40. And then the same for orange, also 20%. So now to do the chi-square statistic, we simply do what our table column headers tell us to do. So you guys probably ought to remember these column headers. You put your phenotype or your possible outcome in the first column, your observed data in column 2, your expected data in column 3, and then observed minus expected in column 4. So in this case, that would be 50 observed minus 60 expected to get negative 10. For the brown row, observed 60 minus expected 40 to get 20. In the yellow row, observed 30 minus expected 20 to get 10. The green row, 40 minus 40 to get 0. And the orange row, 20 minus 40 to get negative 20. The next column header is O minus E squared. So that essentially tells us to get this column, we're going to square the previous column. So in this case, negative 10 squared becomes 100. Don't forget, negative times a negative is a positive. Next one, 20 squared gives us 400 here. 10 squared gives us 100 here. 0 squared, 0. And negative 20 squared gives us 400. Finally, in the last column, O minus E squared over E. So that means take this column and divide it by this column, O minus E squared over E. So in this case, that's 100 divided by 100 here, divided by 60 here to get 1.67. And you could work this in fractions. You're going to see an example in a minute where I work it in fractions. In this case, I decided to go ahead and divide it out. For the brown row, 400 here, divided by 40 here to get 10. The yellow row, 100 divided by 20 to get 5. This row, 0 divided by 40 to get 0. And then the final row, 400 here divided by 40 here to get 10. Now what you've got to remember is that chi-square is equal to the summation of O minus E squared over E. Notice that the last column in my table is O minus E squared over E. So all I've got to do to calculate the chi-square statistic is add up everything in this row. So that's 1.67 plus 10 plus 5 plus 10. So let's see, that would be 26.67. So my chi-square value, 26.67. Now by itself, that number doesn't really tell you anything. So you've got to compare it to a value called a critical value. Let's backtrack a minute and look back at the question. So we just got our chi-square statistic. That was 26.67. Next, what critical value should you use? To use the critical value, you've got to go back to your table. And remember, this table, this chi-square table, is going to be found on your AP Bio formula sheet. To use it, you've got to know two things. You've got to know what are called your degrees of freedom. Your degrees of freedom are always going to be your number of phenotypes or possible outcomes minus 1. 1 less than your number of phenotypes or possible outcomes. So in this case, we had 5 colors, 5 phenotypes. So our degrees of freedom are 4. We're going to be looking at 4 degrees of freedom. Also, you're going to use a p-value of 0.05. Unless you're told different in AP Bio, always use the p-value of 0.05. And we'll talk more about what p-values mean in a minute. So don't worry about that for right now. Just for right now, degrees of freedom, number of phenotypes minus 1, and your p-value, always use 0.05 unless you're told different. So the value we're looking for is where this column and this row intersect, which is 9.49. So we had a chi-square value of, what was it, 26.67. And we had a critical value, which I'm going to abbreviate as CV, of 9.49. All right, you have to compare those. And in this case, if chi-square is greater than the critical value, which it is in this case, we're going to reject the null hypothesis. Now let's think about what that means. Well, first of all, if the calculated chi-square value is greater than the critical value from the table, you reject the null hypothesis. So let's go back to our question for a minute. Our null hypothesis was that there essentially was no difference between the distribution of M&Ms in the bag we counted and the distribution published by the M&M company. So when we reject the null hypothesis, we're saying, oh, yes, there was a difference. And that difference is significant and not likely to be just random. What I found out with this M&M kind of experiment is that the ratios you get are different depending on the factory the M&Ms came from. Different factories use different ratios. So maybe our ratio here came from one company and our actual M&Ms in the bag, or one factory rather, and our M&Ms in the bag came from a different one. But when you reject the null hypothesis, what you're saying is that, yeah, there are some differences between these pieces of data and they're significant. Something's different here. If on the other hand, let's say that our chi-square value had, or let's say our chi-square value instead of 26.67, let's say it was 6.67. So when chi-square is less than the critical value, you're going to fail to reject the null hypothesis. If you fail to reject the null hypothesis, you're saying that maybe there are some differences, but the differences are small and they're probably due to chance. There's a good probability that the differences are just chance differences. They're really small. There's really not any statistically significant differences between our two sets of data. So let's go back and look at the rest of our questions here. So we got a chi-square. We did a critical value. We rejected our null hypothesis. And remember that when you reject a null hypothesis, that means that there are differences between your two sets of data. In this case, the observed and expected data, and that those differences are significant and probably not due to chance. Something else is going on there. All right, that's our first example out of 3. Before I go any further, are there questions I can answer for anyone? Yes, sir.
I have a question, sir. Okay, so for the p-value and the critical value chart, is that just a standard chart?
Yes, that chart is on your AP biology formula sheet, and you can use it throughout the whole exam. Yeah, and okay, I'm going to show you another table in a minute, a different way to get a p-value. But for right now, keep that chart in mind. You're going to have that chart available to you on every AP exam. Anybody else? All right, so let's look at the second question. So a gambler developed a set of loaded die. Die is, well, it should just say developed a loaded die, because die just means one, right? One dice is a loaded die. If it's loaded, it's weighted away. It's weighted so that it's supposed to land on a certain number or set of numbers more often than normal. So in this case, he developed it to land on 5 or 6 more often than a normal die. So the gambler rolled the die. He rolled it 120 times. And this is what he got when he rolled it. So out of those 120 times, he rolled 18 ones, 23 twos, 14 threes, 13 fours, 25 fives, and 27 sixes. So first of all, we're asked to state a null hypothesis. What you got to think, if he rolled it 120 times, you would expect how many of each number? 1, 2, 3, 4, 5, 6. On a normal dice, it should be random, right? So 1/6 of the time, you ought to get ones. So you ought to get 20. 1/6 of the time, you ought to get twos. 20. 1/6 of the time, you ought to get threes. 20, 20, 20, 20, all the way down. you would expect to get 20 of each. Our null hypothesis is going to be that there's no statistically significant difference between the expected values and the values we obtained, the observed values. So we could state it in this case, there's no significant difference between the values we expected and the values we observed. Or we could say there's no statistically significant difference between the number of 1 through 6s that we obtained and the number we expected. Any way you want to state that there's no difference between what we got and what we thought we would get. That's your null hypothesis. Next, we're asked to perform a chi-square analysis on this data. So I'm going to go to the next slide. And here I've written in the observed data. So when we rolled the dice, we got 18 ones, 23 twos, 14 threes, 13 fours, 25 of these, and 27 of these. So to get those expected data, we will add up this first column. That adds up to be 120 total in that column. And then we know we're expecting 1/6 of each number every time we roll. So 1/6 of 120 is 20. We would expect with a normal fair die to get 20 of each out of 120. Now we fill in our table just like we did in the last example. 18 observed minus 20 expected to get minus 2. 23 observed minus 20 expected to get 3. 14 minus 20 to get minus 6. 13 minus 20 to get minus 7. 25 minus 20 to get 5. And 27 minus 20 to get 7. Now we're going to take this column and square it to get the next column. So -2 squared is 4. 3 squared is 9. -6 squared, again, negative times a negative, positive 36. -7 squared, 49. 5 squared, 25. And 7 squared, 49. All right, finally, we're going to do the O minus E squared over E column. So in this case, that's going to be this column divided by this column. Since these all have 20 as the expected value, I'm going to work this in terms of fractions. You could divide it out in a decimal. That would be fine. But I think in this case, fractions are going to make this one easier. So this is going to be 4 over 20. This next one's going to be this column, 9 divided by 20. 36 here over 20. 49 here over 20. 25 here over 20. And finally, 49 over 20. And remember that your calculated chi-square statistic, chi-square value, is equal to the summation of O minus E squared over E. So that tells us to add up this column. So I'm going to add up my fractions here. So let's see, that's 4 plus 9, 13, plus 36 is 49. 49 plus the next 49 is 98. Plus 25 plus 49. So it's looking like that adds up to 172 over 20. Here's where I'd go ahead and divide it out. And again, you could have worked the whole thing in decimal, get the same answer. But I get 8.6 as my chi-square value, 8.6. So let's remember that. I'm going to go to the next slide. Here's where I already worked it out. And now let's go and look at our questions. So our null hypothesis was there's no statistically significant differences between the observed and expected data sets, or the loaded die had no effect on the distribution of numbers obtained when rolling the die. We could write that null hypothesis either of those ways. Perfect. Next, we were asked to determine our expected values, which we did. We took our total number of rolls, 120. We divided that by 6 to get 20 expected for each one. We filled in our chi-square table. We ended up with a chi-square statistic of 8.6. Next, we need to find our critical value. To do that, let's go back to our table. We had 6 numbers, right? 6 phenotypes, 6 outcomes, 1, 2, 3, 4, 5, or 6. So our degrees of freedom are always 1 less than that. So 5 degrees of freedom. In AP Bio, you're going to always use this 0.05 p-value. So you look where those intersect. And they intersect at 11.07. So in this case, we had a chi-square value of 8.6 and a critical value of 11.7. So in this case, chi-square was less than 11 point, it was less than our critical value. And when that's true, you fail to reject the null hypothesis. Now let's think about what that means. And let's look for a second. So this says our chi-square value is higher than our critical value, and that is not. So that's this, it's lower. So we're going to fail to reject the null hypothesis. Now let's think about what that means. When you fail to reject, that's saying that, let's go back to our data table. Here's our actual data. It looks like there's some differences, right? It looks like this column's a little bit different than this column. But when we fail to reject our null hypothesis, we're saying that, yeah, there probably are some differences between those numbers, but they're really small, and more than likely they're due to chance. So what that tells us is that the loaded die the guy made, it's not really getting the job done. It's not causing a big enough difference to be statistically significant. Let's go back at our questions for a second. So we failed to reject the null hypothesis, and again, that means that our numbers, our observed and expected numbers are a little different, but they're not different enough to be considered significant. The loaded die in this case didn't really work. Remember that when your critical value is greater than, let me put it this way, when your chi-square is greater than your critical value, you're going to reject the null hypothesis. When your chi-square value is less than the critical value, which it was in this example, you're going to fail to reject. When you fail to reject, that's saying that there's no significant differences between your observed and expected data. When you reject, that's saying that there are significant differences and that something else is going on. All right, that's example 2. Questions on that one? All right, one more quick example then. Take a minute and read this slide. And I'll read it with you. So a researcher found that onion root tip cells spent 90% of their life cycle in interphase. That's the part of the cell cycle in between cell divisions. And 10% in mitosis. That's when the nucleus and ultimately the cell are dividing. He then treated an experimental group of onion root tips with lectin. So this is a chemical that is supposed to cause the cells to divide faster. When treated with lectin, he found that 2,060 of the onion root tip cells he was looking at were in interphase, and 240 were in mitosis. State a null hypothesis for this experiment. So remember, our null means no. So we could say that lectin has no effect on the number of cells in interphase and mitosis. Or we could say there's no difference between the observed number of cells in interphase and mitosis in the untreated group compared to the lectin treated group. Whichever way you want to say it. Let's look at how I wrote it here. So I wrote lectin has no effect on the cell cycle of onion root tip cells. That works. Next, we're asked to perform a chi-square analysis on the data. So we wrote in our observed values. 2,060 were seen in interphase. 240 were in mitosis. To get these expected values, we're going to add these guys up. So 240 plus 2060, 2300 total cells is what we observed. We're then going to go back and take these percentages. So normally 90% of cells are in interphase and 10% are in mitosis. So we take our total number of cells, 2300 times 0.90, 90%, and that gives us 2070. We take our total number of cells, 2300, multiply that by 0.10, 10%, to get 230. And then, just like we've done in the other two examples, we fill in the table. So O minus E, 2060 minus 2070 to get negative 10. O minus E, 240 minus 230 to get positive 10. Square this column to get this column. So negative 10 squared to get 100. 10 squared to get 100 here. And then O minus E squared over E. So this column divided by this column. So in this case, this would be 100 divided by 2070 to get 0.0483. And in this case, 100 divided by 230 to get 0.4348. So our chi-square value is the summation of O minus E squared over E. So that means add up this column. And let's see if I can do that here in my head. So that's going to be 0.47. Nope. Let's just do it over here. So when we add those up, 0.4831. Our chi-square value is 0.4831. So now we need to compare that to a critical value. Let's go back to our table. Oops, right there's where we need to be. Remembering that our calculated chi-square value is 0.4831. And our critical value in this case, we only had two possibilities, right? Interphase or mitosis. So that leaves us with 1 degree of freedom, one less than our number of possible outcomes. 0.05 is our probability level. So 3.84 is our critical value. So in this case, our chi-square value is less than our critical value. And in that case, we're going to fail to reject the null hypothesis. What that means in this case, there's no statistically significant difference between the number of cells in the control group and the number of cells in the experimental group. Essentially, it's telling us that lectin didn't do anything in this case. Now, if on the other hand, let's say our chi-square value had been 8.48. Our critical value is still 3.84. In this scenario, chi-square is greater than the critical value, which means you're going to reject the null hypothesis. And when you reject it, that's saying that there is a significant difference between your observed data and your expected data, and that something is causing those differences other than chance. So if this had happened in our experiment, that would tell us that more than likely lectin is having an effect. But what we got when we actually looked at our data, we had to fail to reject, which is telling us there's no significant difference between our observed and expected values. All right, that's example 3. Let's see if there's any questions we didn't answer. So we came up with a null hypothesis, no difference between our observed distribution of cells and our expected. We calculated a chi-square value. We found the critical value in the table. We failed to reject our null hypothesis, telling us that there was no significant difference between the distribution of cells in the observed data and the expected data, which tells us essentially that lectin wasn't having the effect we thought it would. All right, I've got one other thing I want to talk to you about, and that is using p-values to interpret a chi-square statistic. So instead of ultimately just going to the 0.05 probability level, we can calculate a p-value or find it another way. So let's just look at what it says up here on the screen. So the p-value is the probability that the differences you see between your observed and expected values are due to chance. So if there's a p-value of 0.05, that means there's a 5% chance that the differences you saw between your observed and expected data are random. There's a 95% chance that the differences are real and due to chance. Scientists usually say any p-value 0.05 or less are significant enough that you reject the null hypothesis. So anytime you have a p-value of 0.05 or less, you're going to reject the null hypothesis. That's important to know. So anytime the p-value is 0.05 or less, reject the null. That means that you want to make sure that the probability that your differences are due to chance, you want to make sure that probability is always less than or equal to 5%. So let's use a different way to interpret our chi-square statistics. Let's go back to problem 1. And remember that in problem 1, we calculated a chi-square statistic of 26.67. And we had 5 phenotypes, so 4 degrees of freedom. Let's look at a slightly different kind of table here. So in this case, to use this table, you're first of all going to go to your degrees of freedom, which is in this experiment was 4. And you're going to try to find your calculated chi-square statistic within this row. So our chi-square statistic was 26.67. So it would have been somewhere way over here, right? Past 13. And then we would go up, and from this row, read the p-value. Notice that as we move from left to right, the p-values get smaller and smaller. So here p-value is 0.01, which means our p-value would be less than 0.01. So what we're saying is we have a p-value less than 0.05, which again tells us that we need to reject the null hypothesis. So this is just another way to interpret the chi-square statistic that you calculated. Let's go and look at example 2. So in example 2, we calculated a chi-square value of 8.6, and we had 6 possible outcomes, which means 5 degrees of freedom. So let's go back to this other table right here. Remember, we're going to go to our degrees of freedom row, which in example 2 was 5. And we're going to try to find our calculated chi-square statistic in this row. Well, our chi-square statistic was 8.6, which means it's somewhere right in here. We then read up to find out what the p-value would be. So our p-value is somewhere between 0.2 and 0.1. It's a p-value that's greater than 0.05. Remember, when p-values are greater than 0.05, you're going to fail to reject the null hypothesis. And again, what that means in example 2 was that, yeah, there were some differences between our data, but the differences were small, and there's a good probability they were due to chance. There's like a 15% chance in this case that the differences were due to chance. All right, let's look at the last one, example 3. So on example 3, we calculated a chi-square value of 0.4831. We only had 1 degree of freedom. So let's go back to our chart. One degree of freedom. Try to find our chi-square value of 0.4831. Scroll over. So 0.48 would be somewhere in here. Point being is that that p-value, once again, is greater than 0.05. And whenever p is greater than 0.05, you're going to fail to reject the null hypothesis. Again, that means that there may be differences between your observed and expected data, but those differences are pretty small. And there's, in this case, almost a 50% chance that the differences happened simply due to chance. That lectin wasn't causing those differences. The differences were due to chance. All right. That's it for our review of chi-square and hypothesis testing. I hope that helped you. And we'll do this again next week. Next Thursday at 10 o'clock, we'll be looking at biochemistry at that point in time. Hope to see you there.